Hi guys, so today we'll be discussing DNA replication within both prokaryotes and eukaryotes, as well as the different mechanisms and processes the cell types utilize in order to ensure that any mistakes that occur during DNA replication, especially mismatches or mutations, are corrected. And so I'm going to begin with a baseline context that we'll need to know in order to advance in this discussion. So first off, DNA within both prokaryotes and eukaryotes exists as a DNA double helix. Each strand held together by hydrogen bonds between the bases, and each strand is anti-parallel to the other strand. So for example, here you have one strand running from the 5' end to the 3' end, and the other from the 3' end to the 5' end. And so before we can even begin DNA replication, essentially what needs to happen is these individual strands that form the double helix must be unwound or dissociated from one another. And the reason that is because our replication enzymes need to access each individual DNA strand in order to use it as a template for replication. And so if they're not unwound, there is no way these enzymes can access and bind to each individual strand, and therefore DNA replication cannot occur. So the enzyme that is responsible for this unwinding or this dissociation and essentially exposing each strand to these enzymes is an enzyme known as DNA helicase. And so DNA helicase essentially comes in, binds to the DNA helix, and starts unwinding the strands and exposing them. And so this process can be seen here in this box for both prokaryotes and eukaryotes. As can be seen here, it's actually a bidirectional process. So you have a helicase binding on one side and moving in this direction, and another helicase that binds here and moves in the other direction. So it's bidirectional. And the same process happens for eukaryotes. And the reason why this is known as the origin of replication is because, as we said, this unwinding exposes the strands to the replication enzymes. The enzymes can then come in, bind to the strand, and carry out their function of creating the complementary base pair. But you may notice that eukaryotes here have multiple origins of replication, while prokaryotes have a single origin of replication. And this is simply due to the fact that eukaryotes typically have larger chromosomes. By larger, I mean more base pairs. And so in order to effectively and efficiently carry out DNA replication, you're going to need multiple exposed regions or multiple replication forks to where the enzymes can come in and replicate the complementary strand from the template strand. So that's all that is. And we also need to talk about the semi-conservative model. Essentially all that is saying that any new double helix strand that originates from another strand will only be 1/2 new. So only one of the strands will actually be newly synthesized. And we can follow this logically from, if we look at this 5' strand, 5 to 3 strand up here in red. Well, if we are using that as our template, if our enzymes are binding to that strand after DNA helicase comes in and unwinds it, well, therefore, that strand will survive and it will be used as a template and will exist in our new daughter DNA strand. So this new daughter strand is actually only 1/2 new. So now that we have DNA helicase that comes in, unwinds the DNA helix structure, and essentially opens up each strand for replication, now we can actually begin our DNA replication process. So a few enzymes that are important to note here. First, we have DNA polymerase 3. DNA polymerase 3 is essentially the primary replicating enzyme. And what I mean by primary replicating enzyme is that this enzyme is the enzyme that is actually responsible for creating the complementary base pair. So it comes to the original strand or the template strand, binds to it, and begins the process of replication. Our polymerase 1, on the other hand, is actually more of a cleanup function, which we will discuss later. and the polymerases 2, 4, and 5 are involved in the DNA repair mechanisms. And polymerase 1, 2, 3, 4, 5, these are the corresponding polymerases for prokaryotes. For eukaryotes, these polymerases are actually called polymerases alpha, beta, gamma, delta, and epsilon. The functions are slightly different, and I'll discuss them after I go through the prokaryotic versions. And so what's important to know for polymerase 3 here are two things. One, it only operates in the 5' to the 3' direction. And two is that it requires a docking site, what I will call a docking site. And we can make sense of the second stipulation, or the second rule for polymerase activity, by just imagining a strand. I'm going to draw it here in orange. So say this is our 5' end, this is our 3' end, and we want to make a corresponding 5', 3' template or replication. Well, in order to essentially latch on, it needs something already there. It needs like a docking site in order to come in, latch on, and then start making the replication strand, and then making complementary base pairs. If there's nothing there to dock on, well, it can't even start making this new strand. And so what this docking site actually is called RNA primer. And so we can see here something called primase, and this is the function of primase. Primase comes in, creates an RNA primer, which is just the RNA complementary base pairs to the template strand. And then once this is laid down, then polymerase 2 can come in, dock onto this RNA primer, and then move in the 5 to 3 direction. So unless primase is there to lay down the RNA primer, polymerase cannot function. So now let's consider the first rule here, where it only moves in the 5' to 3' direction. What this results is in two different strands, one called the lagging strand and one called the leading strand. And we can make sense of this by the direction of the replication fork opening, or the helicase movement. So here we can see the helicase is operating in this direction. So if we can imagine in the leading strand, if we have a RNA primer here, well then, since our new complementary strand is moving in the 5' to 3' direction, since the 5' to 3' direction is the same direction as the opening by helicase, well then, once we are on this RNA primer, the polymerase can simply move and zip along in the same direction as the opening of the replication fork by helicase. So therefore, it's just trailing right behind the opening at the replication fork. However, if we consider the lagging strand, well, the template strand here is 5 to 3'. So therefore, our newly replicated strand is moving in the opposite direction. This is the 3' end, and here would be the 5' end. So consider that polymerase can only, or polymerase 3 can only move in the 5' to 3' end. So if we have an RNA primer here, and our polymerase moves in this direction, well then, in order to catch up to the newly opened, the new openings at the replication fork, well then we're going to need to pack up, make a new RNA primer, and then polymerase 3 needs to move back here and then cover here. And then the same process has to happen over and over, where we're constantly laying a new RNA primer and then moving in the 5 to 3 direction. So let me just erase this right here so you can look at it. So there's actually two ways we can distinguish the lagging strand from the leading strand. So in one way, we're going to have something called Okazaki fragments in the lagging strand. So as I said, since we're not moving in a continuous fashion, we're not just continually making a template strand, and we're constantly having to make new RNA primers and then move in the 5 to 3 direction and lagging behind the opening at the replication fork by helicase, well then we're going to end up with fragments. These are known as Okazaki fragments. And secondly, if you notice, theoretically, in our leading strand, we only need to have one RNA primer. So once we have that RNA primer set initially, we don't ever have to move back and make a new RNA primer. So we just zip through. So we only have one RNA primer. However, in the lagging strand, you can notice we have multiple RNA primers. So those are two ways you can distinguish them. So once polymerase III creates the complementary pair, we have something called DNA ligase. And ligase essentially just glues together these Okazaki fragments. And then we discussed polymerase I, which has a cleanup function. And essentially what it does is it comes in, it recognizes the RNA primers and replaces the RNA primers with the actual DNA complementary base pairs. And essentially what that means is just replacing uracil with thymine. And so another thing to note that's important here is that with each successive replication step, we actually decrease the length of our DNA helix. And the reason why that is consider right here, we have our replication fork. So these have already been, this side has already been replicated. So, and this replication is moving from the 5' end to the 3' end, since it's anti-parallel to the template strand. Well, in this case, right, we're moving 5 to 3', the polymerase can just move normally, no problem. However, the issue results in the other direction. So in the other direction, we're moving 3' to 5', right? So we're going to need our RNA primer. And then once our RNA primer is set, then we can move on and move in with Okazaki fragments. However, consider the case where our RNA primer, which is approximately 11 nucleotides long, consider the case where it's too long in order to fit in the remaining section of the complementary pair. So say we only have 8 nucleotides left to finish the process of replication. Well, our primer is 11 nucleotides long, so it just simply does not fit. You cannot create the entire primer in this region. And therefore, if you have no primer, your polymerase III has no docking site. If it has no docking site, then you cannot create the complementary Okazaki fragment. And so therefore, you're going to lose out on that part of the DNA. It's just not going to be replicated. So you might be thinking, isn't that alarming? Aren't you constantly losing parts of your DNA? When that lead to devastating effects and cutting out genes or affecting genes? Well, the body actually accounts for this with structures called telomeres at the end of the chromosomes. And telomeres are essentially just repeats, non-coding repeats of C's and G's. And they don't actually code for anything. So with each successive replication, we can lose these non-coding nucleotides and nothing will be affected. And the reason why they're C's and G's is simply because they have 3 hydrogen bonds, and since they're located at the end of the chromosomes, they sort of serve as a knot, essentially preventing the chromosomes from unwinding. You can kind of consider them as knots. And so lastly, to bring it to the polymerases in eukaryotes, alpha, beta, and delta are involved in the actual creation of the replication strand, while gamma is involved in replication of mitochondria, and beta and epsilon are involved in the DNA repair mechanism. So let's discuss one replication repair mechanism. So this occurs whenever you have an issue with the DNA base pairing. So for example, polymerase III may put the wrong nucleotide. Instead of putting, for example, C to G, you might put C to A. So we have one type of repair mechanism known as a mismatch repair mechanism, which actually occurs during the replication phase. So that's important to note. And the reason why this can happen is because polymerase itself has a proofreading capability. And so let's consider the template strand A, T, C, G, A, for example, and the new strand T, A, G, C, and then say here it's C instead of T. It should be a T. Well, our polymerase can then bind, move down along, and then recognize a site where you have the nucleotide mismatch. Well, you may be wondering, how does the polymerase distinguish between the correct nucleotide here and the wrong nucleotide, cysteine? And so the way it's able to distinguish between them is through this idea of the hemimethylated DNA. Hemimethylated DNA occurs because the newly synthesized strand has not had enough time within the body to have the modifications. And one of the modifications, among many, is called methylation. And so it has not had enough time to have methylation occur. And these methylations occur at specific sequences known as GATC sequences. And so, say further down along on this original template strand, you have GATC with a methylation. Well, the polymerase is able to recognize that the strand with the methylations is actually the template strand. So this is the correct nucleotide, and so then it will know that this C here needs to be removed and replaced with a thymine. So the next two repair mechanisms that we'll discuss are actually not in the replication phase. They're not in the S phase. They're actually in the G1, G2 phase of the cell cycle. And this repair mechanism essentially locates something called a helix distortion mutation. And one example of a helix distortion mutation is you have UV radiation, the DNA is exposed to it, and two thymines next to each other can actually form a thymine dimer. And this causes a kink in the DNA conformation. And so something called a excision endonuclease can come along, essentially find these helix distortions, excise them, aka remove them, and then DNA polymerase can come in and then fill in the same nucleotides. So you're just replacing the nucleotides after excising them, and then as we talked about earlier, ligase will fill in the little nicks, essentially gluing together the new fragments. And so again, in this case, we are in the G1, G2 repair, in the G1, G2, and we are replacing the nucleotides with the same nucleotides due to a helix distortion. And the last one, which is also in G1, G2, is called the base excision repair mechanism, BER. So in this case, we have a non-helix distorting mutation. So therefore, we cannot use the excision endonuclease. And so we have to use a different mechanism. So an example of this sort of non-helix distorting mutation is cysteine being deaminated to uracil. As you can tell here, this NH2 is essentially replaced with a carbonyl. And as you can tell here, here's guanine. It's supposed to be bound to a cysteine. Well, in the presence of heat or other chemical agents, this cysteine can be deaminated into uracil. But we don't have a helix kink, as in the case of the thymine dimer. And so what happens is this is actually picked up by something called DNA glycosylase. And DNA glycosylase breaks the N-glycosyl bond between the sugar and the base pair. And so essentially we just have no base pair here. And this is called an AP site, essentially apyrimidinic site, just meaning that the purine or the pyrimidine, the base, has been removed. This is then in turn recognized by something called an AP endonuclease. The endonuclease can then cleave it, and then the polymerase 1 will come in, find this cleaved site, and then replace it with the appropriate cysteine in this case. And then DNA ligase will come in and, of course, glue in the ends. So to kind of summarize everything we talked about in terms of the repair mechanisms, we have the first repair mechanism, which is actually during the S phase, or the replication phase, and this is brought about by the actual polymerase. And then we have a second G1, G2 repair mechanism known as the nucleotide excision repair mechanism. And this results from a recognition of helix distorting mutations. And lastly, again in G1, G2, we have the base excision repair which is a non-helix distorting matrix, or distorting mutation. And so that's a summary of the repair mechanisms as well as the replication mechanism. If you have any questions, please comment below and I'll try to answer them. Thank you.